Good day, learners. This is Dr. Francis Ian Salaver, and this is the continuation to Introduction to Immunology. In the previous lecture, we have already discussed the three of the white blood cells, and they are our granulocytes. We have discussed about neutrophils, eosinophils, and basophils. So, we will just have the continuation of the discussion on the two agranulocytes, the monocytes and lymphocytes. We will start first with monocytes. Monocytes are the largest white blood cells. They have diameters that can range up to 20 micrometer. Their size is 3 times bigger than that of our red blood cells. Red blood cells are known to have a diameter of 7 micrometer. So you can fit equivalent of 3 red cells within the cytoplasm of our monocytes. Neutrophils, eosinophils, and basophils usually have diameters of 15 micrometer. Monocytes are easily identified by the presence of kidney bean-shaped nucleus. So take a look at the nucleus of the monocyte in the picture. Can you appreciate that it has that small indentation, making it appear like a kidney bean or a horseshoe? Please take note that according to our first PPT, that neutrophils undergo apoptosis 1 to 2 days after they are recruited from the blood vessel towards the site of infection, and they will be slowly replaced by monocytes. So the second cells that can reach the site of infection is the monocyte. Monocyte is also a phagocyte similar to neutrophils. Just like any blood cell, monocytes are derived from the bone marrow. They will leave the bone marrow and circulate in the blood. And in the blood, we will call them as monocytes. But some of these monocytes will leave the blood and migrate and reside in the tissues. And once they are found in the tissues, you will now call them as macrophage. So macrophages are differentiated forms of monocytes. If you're looking at a peripheral blood smear, you identify the cell as monocyte. But once you're checking on a tissue, you should name the cell macrophage. Macrophages in different tissues have different names. So it's very important for you to memorize their names. In the liver, we call them Kupffer cells. In the central nervous system, we call them microglia. In the glomerulus or the kidneys, we call them as mesangial cells. So it's very important not to be confused between microglia and mesangial cells. Glomerulus, mesangial cells. In the bone, we call them as osteoclast. In the lungs, we have alveolar macrophages or dust cell, dust, D-U-S-T. So I think it's very easy for you to memorize that dust cells are found in the lungs because we sometimes inhale dust in the air that we take into our respiratory tract. In the skin, we call them Langerhans cells, while in the lymph nodes, we call them as follicular dendritic cells. I don't have a copy of the names of the tissue macrophages right now. So what am I trying to tell you? If I was able to memorize their names, you should also memorize the names of the different macrophages in the different tissues in our body. So these are the names of the different macrophages found in the different areas in the body. So I will give you 10 seconds to browse through them. You can even pause this video so that you can memorize the names. Please don't proceed to the next part of the discussion without memorizing the names of the specific macrophages of our tissues. Shown in the photo is one monocyte and one neutrophil. This is a neutrophil. This is a phagocyte. And it is good in digesting the engulfed material because it is provided with enzymes present in its granules. That's why neutrophil is good in engulfing or phagocytosis, and it is also good in killing. Monocytes are better than neutrophils in terms of phagocytosis, so they get to eat more or higher amount of foreign materials compared to neutrophils. But the problem is monocytes are agranulocytes, meaning they lack granules in their cytoplasm. With the absence of granules, it follows that they don't have powerful enzymes similar to that of neutrophils. So the next question now is, why do you think the neutrophils are best replaced by monocytes, when in fact monocytes do not have granules that will help them digest the material that they have engulfed? Yes, they do have lysosomes, but lysosomes may not be enough to digest the engulfed material. Neutrophils have both lysosomes and powerful digestive enzymes in their granules. So the question is, why replace a cell that has a good killing and phagocytosis activity to a cell that cannot digest the engulfed material using enzymes in granules? Monocytes are important in immune responses even if they don't have granules or enzymes that can make them efficient killing machines. They participate in immune responses by acting as antigen-presenting cells. And when you say antigen-presenting cells, they will sample the antigen, take a part of that antigen, display the antigen on their surface, and present it to another cell. And that another cell is a T cell. If the T cell will recognize the antigen presented by the monocyte or the antigen-presenting cell, the T cell in return will activate B cells. B cells now will transform into plasma cells, and plasma cells will produce antibodies. You see, monocytes may not be able to kill pathogens as efficient as neutrophils, but they initiate the process of antibody production by acting as the APC or antigen presenting cell. So take a look at the picture of this macrophage or monocyte. As you can see, this monocyte or macrophage is currently engulfing a pathogen. Once inside the cytoplasm of the monocyte or macrophage, the macrophage will select the most antigenic portion of the pathogen. And what will happen is that antigenic or immunogenic portion of the pathogen will be displayed on its surface, the one shown in the yellow square. And what will happen is this macrophage now will present the antigenic or immunogenic portion of the pathogen to a T cell, particularly a T helper cell. And this T helper cell will act on B cell, and B cell will become the plasma cells, which will later produce the antibodies. This T cell is named T helper because it will help the B cell become plasma cells. Again, you have here a picture of a monocyte or macrophage. Please watch this animation. As you can see, the pathogen was engulfed by the antigen-presenting cell, the monocyte or the macrophage, and a certain portion of the pathogen was displayed on the surface using the MHC protein. After displaying the antigen on the surface using the MHC protein, the APC now, the monocyte, the macrophage or the antigen-presenting cell will now be able to present the antigen to T helper cell. Let's apply what you have learned on this picture. You have an invading microbe that is engulfed by the monocyte, macrophage, or the antigen-presenting cell. A certain portion of the microbe will be displayed on the surface of the monocyte or macrophage, and the antigen will be presented to T helper cell as shown on the green rectangle. What will happen next is the activated T helper cell will interact with B cell, and this B cell will be differentiating to form the plasma cells, and the plasma cells now will produce antibodies. For you to understand the importance of antigen presentation, which will lead to antibody production, let's go back to this particular picture. It's very obvious in the picture that only 1 out of the 4 bacteria was captured and phagocytosed by the monocyte or macrophage. So the other 3 are still free to continue the infection. But after the antigen of the bacterium was presented to T helper, and after the T helper caused the B cell to become plasma cell and produce antibodies, the other 3 bacteria will be killed by the antibodies produced by the plasma cells. It would only take 1 for everyone to suffer. You see, the monocyte magnified the amount of immune response that we created against this particular pathogen. You have learned from the previous discussion that what will help antigen-presenting cells display on their surface the antigen to their corresponding T cells, they will use MHC proteins. MHC stands for Major Histocompatibility Complex. They are named such because they are used in establishing compatibility between donor of organ and the recipient of the organ. Now, there are 2 types of MHC proteins. We call them as Class I and Class II. In the discussion of the macrophage and monocyte as antigen-presenting cell, we actually discussed the Class II MHC. Now, remember, in the previous discussion, it was shown there that the antigen was engulfed by the monocyte or macrophage. So meaning they were taken from the external environment or from the environment outside the cell. So therefore, the antigen that is presented using Class II is extracellular. Please take note of that. Class II is suited for the display or presentation of extracellular antigens. Now, these Class II MHC are present in 2 types of cells. You have the monocyte or the macrophage and B cells. So later on, I will also teach you that B cells can act as antigen-presenting cells, but I will discuss that later. For now, let's focus on the monocyte and the macrophage. Okay, so the monocyte and the macrophage already engulf the pathogen, the extracellular pathogen, and they have already sampled the most antigenic and the most immunogenic portion of that pathogen, and they will load that portion of the pathogen to MHC class 2. And the antigen now will be recognized by a T cell. And a T cell that can recognize class 2 is a T cell that must have CD4 in its surface, CD4 marker. And the T cell with the CD4 marker is T helper. Extracellular antigens are presented using class 2 and they are recognized by a CD4 positive T cell, and that is T helper cell. So to make you understand the characteristics or descriptions of MHC class 2, take a look at this picture. You have there a microbe. It's very obvious the microbe is extracellular. It has to be engulfed by the monocyte or macrophage. Inside the cytoplasm of the monocyte and the macrophage, a certain portion of the microbe will be chosen, and it will be loaded on that green-colored MHC protein. And since the antigen is extracellular to begin with, that MHC should be class 2, and that's the one labeled in the photo. Then, you have their MHC class 2, and then you have there the displayed antigen. And the antigen now will be recognized by a T cell. And, as you can see, the T cell in the picture is labeled T helper cell. It's because class 2 MHC will only be recognized by a T cell that has CD4, and that is T helper cell. And what will happen now? T helper will activate B cell. B cell will become plasma cells. Plasma cells will produce antibodies, and the antibodies will attack the other microbes. Okay. The antigen presentation process is a complicated one. It involves a lot of receptors, and I will be naming them one by one, and I want you to listen very carefully. This is your antigen presenting cell. and it has sampled an extracellular antigen, so it will use MHC class II, the green colored figure in the photo. Loaded on the MHC class II is the antigen. Take note, the antigen will be recognized by the T cell receptor, the red colored figure. And part of the T cell receptor is CD3. That's a marker of your T cell. What will recognize if the antigen is presented using class 1 or class 2 is now the CD4. Antigen is recognized by CD3, which is part of the T cell receptor, while the MHC class 2 is recognized by CD4. Let's apply that one on this figure. This is the antigen presenting cell, and it has sampled an extracellular antigen, so it will use MHC class 2, and this is now the antigen being presented, and that antigen will be recognized by the T cell receptor, the blue colored figures there. Part of them is the CD3, and what will recognize MHC class 2 is CD4. Can you appreciate that the CD4 particularly interacted with the MHC class 2 and not with the antigen? Let's have a summary. If the pathogen is extracellular, then it will be taken up by the monocyte or macrophage. The monocyte and the macrophage will act as antigen presenting cells. They will use class 2 to present the antigen of the microbe because it is extracellular. So what is activated is T helper, right? And T helper is known to help or activate B cells to form plasma cells. And plasma cells, so let's have this fill in the blanks. Extracellular antigens are presented by APCs using MHC class 2. Okay. The presented antigen is recognized by the T cell receptor which contains CD3. The MHC class 2 is recognized by the T cells CD4 marker. Very good. You have learned a while ago that aside from monocytes and macrophages, B cells can also present antigens using class 2. Can B cells really present antigens to T helper cells? No. B cells are known to have surface antibodies or surface immunoglobulins, and we will discuss that later on. I want you to take a look at the B cell pointed by the red arrow in the photo. Appreciate that one of its surface immunoglobulins has captured a specific antigen. Do you know what will happen? After the binding of the antigen to the surface antibody, the antigen will be internalized by the B cell as shown in the picture. After the internalization of the antigen, the B cell will present the antigen using class 2. Doc, why class 2? Because the antigen is extracellular as shown in the photo. And you know what will happen? The B cell now using the class 2 will present the antigen to T helper cell. The question now is, if B cell can present an antigen, what is now the importance of macrophage? If the B cell can just directly activate T helper and the T helper will activate B cell to become plasma cells and antibodies will be produced. So therefore, we can remove the antigen presenting cell or the macrophage or monocyte from the picture because B cell can already present the antigen on its own. So as shown in this picture, you have here a macrophage that is acting as antigen presenting cell. Then you also have here a B cell that is also acting as an APC, and they are presenting the antigens to T helper cells. Who is now better in terms of activating the T helper cell? Is it the monocyte or the macrophage or the B cell? For you to understand that, I want you to imagine a family who is composed of baby B cell, tatay T cell, mama macrophage. So let's say, for example, baby saw the antigen first before mama. So baby thought that she has found her soulmate. Then, baby B cell will present her soulmate to tatay. And baby B cell would say, Tay, please allow us to get married. And tatay replied, Baby, can we wait for your mama to decide also on this matter? As soon as mama has already met the soulmate and mama would also introduce the soulmate to tatay, tatay will now approve the marriage of baby and her soulmate. So the point is, B cell is not as good as activating the T helper cell as the macrophage. So even if B cell can present the antigen to T cell, the T cell will still wait for the macrophage to present the same antigen before the T helper cell will activate the B cell to become plasma cell and produce antibodies. Before I will be able to discuss with you guys the importance of using Class I MHC, let me first describe to you the structures of viruses. Viruses are not cells. They are just basically piece of a microorganism that has DNA and RNA. As you can see in the picture, the virus has a viral genome. So that could either be DNA or RNA, but never both. That viral genome is covered by a capsid, and the capsid is later covered by the envelope. Can you appreciate that the envelope has protein spikes? These spikes will be used by the virus to bind to cell receptors, which will cause them to be internalized by the human cells. What do you think is the purpose for the viruses to be internalized by human cells? Take a look at this virus. It will use its envelope proteins to bind to the receptors present on the surface of the human cells, and that will cause them to be internalized by the target human cell. What's the purpose of them invading the human cell? Because they are not cells to start with. They don't have the machinery for them to replicate themselves. So they need to be found inside the human cells so that they can use the machineries inside the human cells for them to replicate. That's why viruses cannot survive without a host cell. And these organisms, microorganisms, are described to be obligate intracellular. They always have to be found inside a host cell for them to survive. What I want you to learn from that discussion is that, unlike most bacteria, viruses are not considered as extracellular antigens because they are found inside the cell, not because they were engulfed, but because they invaded the cell, and that makes them intracellular. So if the pathogen that is causing us to become sick is intracellular, similar to viruses, the MHC that is best to use to present them is MHC class 1. Now, this MHC class 1 is found in all nucleated cells in the body. Why do you think basically all nucleated cells in the body should contain class 1? Because they are the ones prone to being infected by viruses. So God loves us so much that He provided all of our nucleated cells with class 1 so that whenever they are invaded by viruses, they can always inform the immune system using class 1 that they have a virus inside them. And the T cell that will respond to MHC class 1 is the T cell that has CD8, and that is T cytotoxic cell. So for you to easily remember, extracellular, it should be presented using class 2, and the T cell that is supposed to respond is CD4. 2 times 4 is 8. For intracellular antigens, the MHC of choice is class 1, and the T cell that will react is CD8. 1 times 8 is 8. So it should always be a product of weight. 2 times 4 is 8. 1 times 8 is 8. So you have here a picture of the virus that is invading a human cell. And then what will this human cell do? It will choose a certain part of the virus or any protein produced by the virus, and that cell will load it on the yellow colored or golden colored class 1. And as you can see, the viral antigen along with the class 1 will be displayed on the surface of the virally infected cell. And there's one T cell that will respond to it, and that T cell is a CD8 positive T cell, and that is the T cytotoxic. So let's try to identify the different receptors or markers involved in antigen presentation using class 1. This is your virally infected cell, which is acting as the antigen presenting cell in this case. Then you have here the MHC class 1, the green colored figure. Loaded into it is a viral antigen or viral protein. Same as that in class 2, the viral antigen or the intracellular antigen will be recognized by CD3 as part of the T cell receptor. So meaning in both antigen presentation using class 1 and class 2, CD3 is always involved. What will now recognized if the MHC is class 1 or class 2 is the CD8. So in extracellular antigens, antigen is recognized by CD3, part of the T cell receptor. Class 2 is recognized by CD4. In the case of class 1 MHC, antigen is still recognized by CD3, part of the T cell receptor, but the class 1 MHC is recognized by CD8. And CD8 is present in T cytotoxic cell. So take a look at the maroon colored virally infected cell in the picture. Can you appreciate that it is using that light blue colored MHC class 1 to display the purple colored viral antigen? And the viral antigen is recognized by the T cell receptor, which contains CD3, and the T cell is labeled as CD8. So if the T cytotoxic or CD8 positive T cell will recognize that a cell is virally infected, how do you think it will create the immune response to help the cell with the virus? The T cell now will release two important chemicals. Perforin, from the name itself, it will perforate something. It will create pores on the surface of the virally infected cell. And the pores now will serve as the portal of entry for another chemical, and you call it granzyme. Granzyme will enter through the pores into the virally infected cell, and it will cause the cell to undergo lysis. So the infected cell will look into the eye of T cytotoxic. Hey, I presented the antigen to you because I was asking for help. Why are you killing me? And the T cytotoxic asked the infected cell, where is the virus? The infected cell replied, inside me. So the T cytotoxic replied, how can I kill something that is inside you without killing you first? So if class 2 MHC antigen presentation will result to antibody production, the usage of class 1 in antigen presentation will lead to the lysis of the virally infected cell. So take a look at this picture. You have the infected cell displaying the antigen using class 1 MHC, and the antigen is recognized by the cytotoxic T cell. The cytotoxic T cell number 2 will release perforins, which will create pores on the surface of the virally infected cell, which will cause the entry of granzyme, which will eventually cause the cell to undergo lysis. Fill in the blanks. Intracellular antigens such as viruses and intracellular bacteria are presented using MHC class 1. The presented antigen is recognized by the T cell receptor with the CD3 marker. The MHC class 1 is then recognized by the T cell's CD blank marker. That is what? CD8. Let's have another short quiz. Just answer class 1 or class 2 MHC. Number 1, involved in the presentation of extracellular antigen. Class 2, involved in the presentation of intracellular antigen. That's most likely class 1. Very good. Recognized by CD4 positive T helper cell. That is class 2. And look at how I wrote down the letter H in helper. I just placed there an inverted number 4. That is to help you memorize that T helper has CD4. And then recognized by CD8 T cytotoxic cells. Class 1. I intentionally highlighted the 2 letter O's in there so that if you combine them, they look like number 8. Presentation of antigen using this class of MHC will result to production of antibodies. Class 2. And the presentation of antigen using this type of MHC will result to cell lysis. Class 1. So I hope you were able to get everything correctly. Let's have sample cases. Chickenpox is a viral infection. I had it when I was still a child. So expect that during the time that I was having chickenpox, the cells infected by the virus should have undergone lysis. But how come I can confidently say in front of you that I am already immune to chickenpox because I already have antibodies against the virus? Recall that viruses are intracellular antigens and they're supposed to be presented using class 1. And class 1 antigen presentation should end up with cell lysis. And only class 2 antigen presentation will result to antibody production. So, why do you think I was able to develop antibodies against chickenpox if it is caused by a virus? The answer is, every time a virally infected cell will be killed by T cytotoxic cell or natural killer cell, they will form cellular debris. And this cellular debris would contain dead viruses. This cellular debris will be picked up now by our macrophages or monocytes. So from being intracellular, the dead viruses now will become extracellular because they were engulfed by the monocyte or macrophages. So that's the reason why the dead viral particles or antigens will now be presented using class 2, and it will later result to the production of antibodies against the virus. Now you see the connection and somehow the completion or the linkage between the different immune responses in the body. Do you find it interesting? You have also learned from the previous PPT that eosinophils attack parasites via the process called antibody-dependent cell cytotoxicity, wherein the eosinophil will bind to the antibodies present on the surface of the parasitic worms or protozoans, and then they will release their major basic protein through the process of degranulation. The question now is, these parasites are too big to be engulfed by our monocyte and macrophages. You know very well that for antibodies to be produced, antigen presentation must be initiated first. So, question, who sampled the parasites for them to be presented to T helper if they are bigger than our antigen presenting cells? The answer is, every time that these parasites would wander in our body, they might come in contact with damaging chemicals or enzymes. So, portions of their surface or skin will peel off, and those smaller structures now will be engulfed by our antigen presenting cells. So, you now have antigen to be presented to T helper. That's why there would still be antibody production. With that, the body will be able to generate antibodies against the parasitic worms. These antibodies will bind to the antigens present on the surfaces of the worm, and these antibodies now will activate eosinophils to degranulate and release their major basic protein. And this process is called again what? antibody-dependent cell cytotoxicity. Before you are allowed to go on to your internship, you must have completed your hepatitis B vaccination. Why? Because hepatitis B can be acquired through accidental needle prick, which is possible when you are in your duties. Hepatitis B can cause a chronic form of infection. When you say chronic form, the infection has been there for more than 6 months. The primary organ that is affected by hepatitis B is the liver. And one of the unique characteristics of the liver cells is that they can regenerate and replace damaged ones. So every time a liver cell will be infected by the hepatitis B virus, and if that cell will undergo lysis, releasing now the newly replicated hepatitis B virus, the healthy or remaining liver cells will try to compensate by multiplying to replace the damaged or dead ones. The problem is the infection has been there for more than 6 months. The thing is, every time that the cell will subject itself to replication, it has to duplicate its DNA. The more that it has to undergo replication, the higher the chance that the duplication of the DNA could have any errors. So, the error or the mutation during the DNA duplication will put at risk the newly formed hepatocytes to have mutations, and eventually those mutations can lead to the occurrence or development of hepatocellular carcinoma. See, knowing the principle behind the antigen presentation of class 1 and knowing that hepatocytes will really die after they will be infected by the virus, you can now explain why hepatitis B can lead to its chronic form and can eventually lead to hepatocellular carcinoma. Let's have another case, and this time it's malaria. You know very well that malaria is acquired through the bite of a female Anopheles mosquito. And what is introduced by the mosquito is the stage we call the sporozoite. So just follow the life cycle. You have there the sporozoite infecting the liver cell, and inside the liver cell, they will form, look at number 3, schizons. The schizons will cause the liver cell to rupture, releasing now the individual merozoites in number 4. And these merozoites now will infect red blood cells. And they will undergo another round of schizogony inside the red blood cell. That's why you can see there inside the red blood cell the development of schizon. Now, looking at the picture, I want you to classify Plasmodium species. Are they extracellular or intracellular? Remember, they can invade human cells. The merozoite will invade red blood cells. They were not engulfed by the red blood cells to start with. So, therefore, Plasmodium species are considered to be intracellular or extracellular. They are intracellular. So, knowing these facts, you can say that, okay, if they are intracellular, so at least we can rely on our cytotoxic T cells to destroy the the red cell that is infected by the Plasmodium species, and the infection will be eliminated. But the thing is, people with malaria will never recover without treatment. So if there's a T cytotoxic that can kill intracellular organisms or parasites, then how come the immune system is not working against the Plasmodium species? Recall that Class I MHC is widely distributed in the body, particularly in all nucleated cells. In the case of malaria, the affected cell is the red blood cell. And you know very well, mature red blood cells do not contain nucleus. So it follows that they cannot express Class I MHC. That's why T cytotoxic cells, even if they will bump to a red blood cell with a Plasmodium species inside it, it will never ever have an idea that there's something wrong with that red cell, because the red cell is not a nucleated cell and it cannot present using class 1 MHC. The last case is HIV. You know very well that the retrovirus, the causative agent of HIV, is known to infect CD4 positive cells in the body. So you have here a picture of the HIV virus. As you can see, it has this GP120 protein, the one that it will use to bind to the CD4 receptors in any cell of the body. And you have learned a while ago that T helper cells contain CD4. So they are the target cells of our HIV virus. So once the HIV virus will use the GP120 and bind to CD4, they will be internalized by the CD4 positive T helper cell. Please take note that T cells are nucleated. In the case of HIV, it is the T cell that is infected. For a very long time, these T cells have been protecting you. But in the case of HIV, they are the ones being infected. And since they are nucleated, they will also present the virus using class 1. Yes, they have CD4, I recognize that. But since this time, they are the ones being infected, they also have to call for help. And since they are dealing with a virus, they will use class 1 MHC to send a signal, a message to the immune system, hey, I have a virus inside me. Since the CD4 positive T cell will present the viral antigen using class 1, the T cell that will respond to it is CD8 positive T cytotoxic cell. And you know very well that T cytotoxic T cell is designed to kill virally infected cell. So to people who have HIV and they are not maintaining any medication, their T cytotoxic T cells are slowly killing their CD4 positive T helper cells. And how important is T helper cell again? It's the one that is causing the B cell to produce antibodies. If you remove them from the picture, then the person will no longer be able to produce antibodies. And the person will now have immunosuppression. Do you know that T cells need 2 signals before they can be fully activated? So what are these 2 signals? The first signal is provided by the interaction of the MHC class 1 or class 2 with the loaded antigen to the T cell receptors, which contain CD3 and CD4 or CD8. So I will use MHC class 2 as an example. So this is your antigen presenting cell. This is the MHC class 2 with the loaded antigen. And then this MHC class 2 and antigen will interact with the T cell receptors and CD4. The interaction of all of these structures or receptors will provide the first signal. So the first signal for T cell activation is involving antigen and CD3, class 2 and CD4. That's for T helper activation. It can involve antigen and CD3 or the T cell receptor and class 1 MHC and CD8 interaction for the activation of T cytotoxic cell. The second signal is provided by another set of receptors. So, let's have an example. This is your antigen presenting cell. This is your first signal. The second signal now will be provided by the interaction of, you better listen, the B7 or CD80 or CD86 of the antigen presenting cell and the CD28 of the T helper or T cytotoxic cell. The second signal involves what we call co-stimulatory. What is the importance now of memorizing that there are co-stimulatory molecules involved in the activation of T cells. If the antigen presenting cell in the body will accidentally engulf self-antigen and they will present the self-antigen onto their surface, automatically, due to an unknown mechanism, these antigen presenting cells will lose their ability to express B7. Why do you think na the B7 should disappear or should no longer be expressed? Because presentation of the self-antigen can lead to the production of antibodies that will destroy your own antigens, and that is what we call autoimmune disorder. So, to prevent the possibility of autoimmune disorder brought about by faulty antigen presentation, the antigen presenting cell should be disabled from expressing B7. So in the photo, you have here two T cells. One T cell has both the first and the second signal because the antigen presenting cell, the one labeled as APC, was able to express B7. But if that APC, focus on the right side of the photo, will present self-antigens, it will no longer be able to present B7. And what will happen is that T cell will only receive the one of the signals and it will never reach full activation. If that's the case, the T cell will just undergo what we call anergy. What is anergy? It's a mechanism in which T cells or any lymphocyte in the body is intrinsically, functionally inactivated following an antigen exposure or encounter, but remains alive for an extended period of time but in a hyporesponsive state. And that T cell will no longer be involved in any immune response because once activated, that T cell could cause an autoimmune disorder. So one of the mechanisms on how our body would prevent the occurrence of autoimmune disorder is called anergy. And anergy is characterized by the absence of what? B7 in antigen presenting cells. And that ends the part 2 of my discussion to Introduction to Immunology. Molecule, because they just pair with the structures involved in the first signal to fully activate the T cell. And what are the co-stimulatory molecules? On the part of the antigen presenting cell, you have B7. B7 is also known as CD80 or CD86. While on the part of the T cell, you have CD28 antibodies. So, antigen presentation involving Class II MHC will lead to antibody production, and antibodies will attack the other extracellular antigens not attacked by the or sampled by the monocyte or macrophage. But please take note, antibodies fall under adaptive immunity. So they will only attack the microbe that is similar to the microbe that initiated the antigen presentation and antibody production. But the point is, MHC class 2 antigen presentation will result to production of antibodies. As you can see, it has caught one of the 4 bacteria. And then what will happen is the engulfed bacterium will be digested using its lysosome in the formation of the phagolysosome. I hope you can remember that from histology. After that, a certain portion of the bacterium will be loaded into a protein. That protein is labeled MHC, and we will discuss that later on. So this MHC protein will help the macrophage or the monocyte or the antigen presenting cell to show or display the portion of the antigen on its surface.